Welcome to this class on neuroscience of human movement. So, in this class we will continue our discussion on basal ganglia, specifically motor functions. This class will be focusing, or we will start our discussion on motor functions of basal ganglia. We discussed this in the previous class. So, in the cortex, areas responsible for motor functions, these are primary motor cortex, supplementary motor area, premotor cortex and cingulate motor area. From these areas there is projection onto basal ganglia, specifically the striatum, or in most cases putamen, right. From there the output goes to the pallidum, and from there to the thalamus, where within the thalamus the motor areas project to ultimately the VAVL thalamus, or ventroanterior and ventrolateral thalamus. An important principle is the concept of segregated circuits. In this, inputs received from different areas of the motor cortex, say for example the hand and the leg, are kept separate in the striatum, are kept separate in the pallidum, are kept separate in the thalamus. So, essentially there is some sort of somatotopy, unlike the somatotopy in the primary motor cortex, or unlike the somatotopy in the cerebellum. This somatotopy is not so profound or so well studied. However, there is this separation or segregation of different areas. So, somatotopic arrangement are maintained throughout different structures within the motor loop. So, within the basal ganglia, so there is this separate arrangement that is maintained for different areas, right. This is again within the motor loop. Of course, note different functions are differently maintained. In other words, motor function is separately maintained from executive function, which is separately maintained from limbic function, etc. So, that is different. Within the motor loop, different areas of the frontal cortex, different areas of the motor cortex project to different areas in the striatum, and the connections from the striatum to the pallidum, and from the pallidum to the thalamus, and from the thalamus back to the cortex are maintained separately throughout. There is not mixing of this. And not just that, the thalamus projects to the cortex, that is the major output of the thalamus, but a particular region of the thalamus, the centromedian nuclei of the thalamus, actually projects back to the striatum, right. So, essentially acting as a feedback loop. So, that means the striatum is receiving input not just from the cortex, but also from the thalamus. We discussed this in the previous class. We said that the cortical inputs received by the striatal medium spiny neurons are located in more distal dendrites when compared with the inputs coming in, say from the thalamus, or say from the local interneurons of the striatum, right. So, the dopaminergic inputs coming in from the substantia nigra pars compacta and the cortical inputs coming in from various regions in the cortex are actually projecting to the distal dendrites, whereas thalamus and the local circuit neurons actually project close to the soma. That means they have an ability to nicely modulate, finely modulate the output of the striatal medium spiny neurons, right. So, where does that thalamus neuron originate? In the centromedian nucleus of the thalamus, right. So, also it has been noted that different subloops or different subcircuits exist within the motor loop for planning, sequence coordination and execution. So, that means depending on for what function I am going to use a particular limb or a particular organ of the body. So, depending on whether I want to pick up the pen or whether I want to push the pen out, right, or whether I want to perform a cyclic movement like that, or whether I want to write, for example. Depending on that, these are different functions but essentially we are using the same limb and the same effector for that, right. So, essentially these functions are different. It turns out that basal ganglia has different loops for different aspects of this movement related behavior, right. So, essentially for planning there is a different loop, for sequence related function or sequencing of actions there is a different loop, and for execution of action there is a different loop. So, there seems to be some segregation that is maintained in terms of function also, not just in terms of areas, right. So, of course, hand and leg areas are maintained separately, but within the hand, depending on what I am going to do, depending on whether there is a planning that is required, so that is also segregated. How do we know these things essentially? So, I am speaking as if we know everything. How do we know these things? Actually, our knowledge of basal ganglia function is limited, but still it has improved tremendously in the last few decades, right. Importantly, this has come from two sources. One is electrophysiological recordings. So, what this involves is either stimulating electrophysiologically specific areas in the striatum or specific areas in the basal ganglia and observing the output in terms of behavior, or of course, recording from specific regions of the basal ganglia during natural everyday behavior, for example, or natural lab related behavior. So, whatever. So, this is one way of doing it. This is basically electrophysiology. The other way of doing it is to use anatomical principles. That is something that we will discuss in the next slide, right. So, what is done here in electrophysiology is recording of neural activity during passive and active movements, right. So, when the monkey's arm is moved passively by say a robot, what is the response in the basal ganglia, or when the monkey itself is volitionally making the same movement, what is the activity in a specific region within the basal ganglia, right. What this has shown is that specific area in the striatum is active during movement of specific part of the body. So, essentially for example, leg movements, during leg movements dorsolateral area of the putamen or neurons in the dorsolateral area of the putamen are active, and during facial movements ventromedial or a different region of putamen are active, and during arm movement a specific band or a specific area that lies between the dorsolateral and ventromedial region of the putamen was used for arm movements. Once again, you do not have to remember all the little details of this is dorsolateral, this is ventromedial, and this is the band between the two or this, but it is important to remember the concept. It is important to grasp the concept that the leg movement areas in the striatum is different from the facial movement areas in the striatum, which is different from the hand movement areas of the striatum. You may not be able to remember dorsolateral, ventromedial, right, but it is necessary to remember that these are maintained separately. The other way of studying this, of course, is anatomical evidence, right. How is this done? This kind of work was pioneered by Dr. Peter Strick and his colleagues. So, what they do is they inject rabies and herpes viruses intracerebrally, and it turns out that these viruses can be transported across synapses, right. So, this virus is taken up by neurons and transsynaptically transported, that means that they can cross the synapses. So, but it takes time, it does not happen instantaneously, of course. So, as soon as the injection is given, it takes anywhere between hours to days for these viruses to be transported, right. And it turns out that these viruses show up in specific manner in anatomical slicing. So, what happens is that these viruses are injected intracerebrally. So, basically using injections these viruses are injected into specific regions within the basal ganglia. Once again, you have to know to which area you are injecting it into, and then what you can do is after say 1 day or after 2 days, after a specific timeline, right, you sacrifice the animal and perform histological analysis. Basically, you take specific slices of the basal ganglia circuitry and then identify which areas of the basal ganglia actually show staining for this particular virus, right, showing, then using that you can actually identify which areas of the basal ganglia are connected to which areas of the basal ganglia once again. So, this method helps us to trace the pathways, right, specific circuit path. This is a revolutionary concept. This is also used to trace pathways from other regions to other regions. In other words, this is also used to trace pathways from the spinal cord to the cortex and vice versa, etc., right. Of course, separate injections in primary motor cortex, supplementary motor area, premotor area produces labeling in ventrolateral thalamus and the GPI, right. Using a similar approach, pathways from the cortex to striatum and from striatum to pallidum have also been found and have been found to be segregated, right. So, essentially once again this provides strong evidence for the concept of segregated circuitry. So, again this concept, that this idea that different regions sending inputs to the basal ganglia, there is not mixing of inputs from multiple regions. These regions and these inputs are maintained separately and the outputs are also sent separately. So, there is segregation, but importantly, an important part to note, but an important point to note is that a large number of cortical neurons project to a relatively smaller number of neurons in the striatum, right. So, that means there is convergence, here, and also we said that a relatively large number of the striatal medium spiny neurons project to a relatively smaller number of neurons in the output nuclei, which are basically globus pallidus internal and substantia nigra pars reticulata, right. So, essentially what is happening is one more level of convergence here. So, this would make us believe that, oh okay, so from multiple areas it is projecting to a smaller area. So, that means integration of information from multiple areas in the cortex happens in the striatum. Actually, this is not what happens is that convergence happens within, this very important concept, convergence happens within functional and anatomical areas within the basal ganglia, not between. So, essentially this is done while maintaining segregation. There is convergence. So, a lot of cortical neurons come say from supplementary motor area and they project to a smaller number of neurons in the striatum, and those neurons in the striatum project to a smaller number of neurons in the output circuitry, basically the pallidum, right. Now, if you take similarly a different area, say the cingulate motor area, right, a large number of cingulate motor area neurons project to a smaller number of striatal neurons and a relatively large number of striatal neurons project to a smaller number of pallidal neurons. Segregation is maintained while at the same time convergence is happening. So, this is an important point to remember. So, convergence happens within but not between functional areas of the basal ganglia, thus maintaining the segregation, maintaining the somatotopic integrity. So, in summary, essentially motor loop is the loop from the cortex to the striatum back to the GP, back to the cortex. So, in summary, motor loop is this loop that starts or that originates in the cortex and projects to the striatum and from there it projects to the pallidum, and from there it projects to the thalamus and back to the cortex. Note, this cortex can be any cortex except the primary visual and the primary auditory cortex. This cortex is always the frontal cortex, something to remember, and there is segregation of input and pathways, right, and there is separate subcircuits for planning, sequence coordination and execution, and there is feedback coming in from the centromedian nucleus, and there is feedback coming in from the centromedian nuclei of the thalamus back to the striatum, and there is convergence from the cortex to the striatum and from the striatum to the pallidum, right, and convergence happens within each circuit, but not between circuits. So, with this we come to the end of this lecture. We will continue this discussion in a future class. Thank you very much.